Hello everyone. Today we'll talk about the interactions between the drug and the receptor and the types of the bonds, the intramolecular bonds that occur between them. We'll also talk about the structure-activity relationship, the pharmacophores, isosteres, bioisosteres, and prodrugs. So first, before we start, let me remind you that anything I add to the notes is for your own education. You're welcome to just stick to the handouts and you could skip this video if you want. So let's start by the reading objectives. As you go through the notes, I want you to be familiar with the types of drug targets and their examples, and then understand the chemical nature of the proteins since they are the most common type of drug targets. And then I want you to understand the overall process of the receptor-ligand interaction, which involves forming intermolecular interactions, conformational changes, signal followed by signals transduction, followed by departure of the ligand from the receptor. Okay, so basically the stages of the activation of the receptor, and also differentiate between the binding site for an endogenous ligand and the allosteric binding site. As you will see, there are two binding sites, or not always, but some receptors can have, besides the binding site for the endogenous ligand, some receptors can have something called allosteric binding site. So make sure you know the difference between these two. And then also differentiate between different kinds of intermolecular bonds, the hydrogen bond, the hydrophobic bond, van der Waals, ionic, all these, and be able to rank them by their strength. And then relate the impact of the strength of an intermolecular bond on the affinity of the ligand. Okay, so if the strength is higher for a bond, then the ligand should have more affinity to bind to the receptor. And then how is the affinity measured using the dissociation constant, and what is the relationship between the dissociation constant and the affinity. And then relate the impact of the intermolecular bonds on the induced fit and how this affects the biological activity. Okay, and then also relate the structure-activity relationship to the drug-target interactions and how the pharmacophore is defined. So make sure you know how to. There are like 2 or 3 ways to define a pharmacophore. One of them is the generalized bond-type pharmacophore. And then understand the similarity principle as it relates to the pharmacophore. Finally, differentiate between isosteres, bioisosteres, and value their importance in drug design. And then define what a prodrug is and examples of their uses in improving the feature of a drug. Okay, and as you know, there will be an IRAT, a TRAT in the lecture in the class, and then of course we'll have some TBL applications. And here is some optional readings for you. These 2 books unfortunately are not available in Access Pharmacy, but if you want to read them, I can make copies for you. I have both books. Actually, I have access to both of them, one online and one in my office. And I believe this one here is available at the library too. You can borrow it if you want. Anyways, but the notes should be sufficient. So this is just optional reading for you. Okay.
In order to design painkillers, we need to understand how the pain receptors are activated and how they interact with natural chemical messengers, also known as endogenous ligands. The idea of having a receptor that binds to the natural chemical messenger or to the drug was first developed by Ehrlich, who explained the biological action of molecular entities. He postulated that receptors are certain side chains that are located on the surface of the cells, and these side chains are complementary to the chemical messengers or the endogenous ligands. The two, which is the side chains and the chemical messengers, could therefore, since they are complementary, could therefore interact with one another and produce the biological action. Um, so let's understand these. In order to understand these side chains, let's explain the chemical nature of these receptors, or also known as drug targets. Okay, so drug targets can be classified based on their chemical nature into four groups. There could be lipids, such as cell membrane lipids. There could be nucleic acids, such as DNA or RNA. There could be carbohydrates, like the cell surface carbohydrates and antigens. And last but not least, there could be proteins, which is the most common type of these drug targets. And these proteins could be structural proteins. These are proteins that are involved in building the structure of the biological system, such as tubulin. They could be transport proteins that are responsible for transporting polar molecules or amino acids. They could be also transmembrane, and there are three kinds of transmembrane proteins. There could be an ion channel protein, transmembrane, which means that they are attached to the membrane of the cell, and they have some part of them as outside of the cell and another part inside the cell, so they are known as transmembrane proteins. So they could examples are again ion channels, or the G protein coupled receptor, a very important protein, or receptors that has enzymatic cytostolic domains that could catalyze reactions such as kinases. And they could also be cell surface adhesion receptors. They could be intracellular receptors, and they could be extracellular enzymes, such as the enzymes that catalyze the reactions inside the cells. Such as esterases, for example, which are an example of extracellular enzymes. Since proteins are the most common type of drug targets, we will review the chemical structure of proteins. I know some of you have not taken biochemistry course, and to make sure everybody is on the same page, I provided a video for you to watch. So just click on this video, and while you watch the video, make sure you are able to answer the following questions, which is what roles do proteins play in biological system? What are the building blocks of proteins? What type of atoms are involved in these building blocks? And what is the difference between primary, secondary, tertiary, and quaternary structure of a protein? So let's go ahead and watch the video.
Proteins play countless roles throughout the biological world, from catalyzing chemical reactions to building the structures of all living things. Despite this wide range of functions, all proteins are made out of the same 20 building blocks, called amino acids. The way these 20 amino acids are arranged dictates the folding of the protein into its unique final shape and its function. Amino acids are made of carbon, oxygen, nitrogen, hydrogen, and sulfur atoms. These atoms form an amino group, a carboxyl group, and a side chain attached to a central carbon atom. The side chain is the only part that varies from amino acid to amino acid and determines its property. Hydrophobic amino acids, such as leucine and isoleucine, have carbon-rich side chains, which don't interact well with water. Hydrophilic amino acids, such as serine or threonine, interact well with water. Charged amino acids like glutamic acid or arginine interact with oppositely charged amino acids or with water. The primary structure of the protein is the linear sequence of amino acids as encoded by DNA. The amino acids are joined by peptide bonds, which link an amino group and a carboxyl group. A water molecule is released each time a bond is formed. Specific amino acid sequences give proteins their distinct shapes and chemical characteristics. These protein chains often fold into two types of secondary structures stabilized by hydrogen bonds. A protein chain can fold into a rigid alpha helix, forming regular patterns of hydrogen bonds between the backbone atoms of nearby amino acids. Backbone atoms of the chain can interact side by side to form beta sheets. Many proteins fold into a compact globular shape, with hydrophobic side chains sheltered inside away from the surrounding water. The functions of many proteins rely on this folded structure. For instance, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds oxygen. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The 4 subunits of hemoglobin cooperate so that the complex can pick up more oxygen in the lungs and release it in the body. Many proteins rely on the ability to recognize the shape of specific molecules in order to function correctly. The flexible arms of antibodies protect the body from disease by recognizing and binding to foreign molecules, and thus preventing the viral RNA or DNA to enter the cell. Collagen forms a strong triple helix that is used throughout the body for structural support. The calcium pump moves ions across cell membranes, allowing the synchronized contraction of muscle cells. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood sugar level. Alpha amylase is an enzyme with a catalytic site that begins the breakdown of carbohydrates in our saliva. Ferritin forms a hollow shell that stores iron from our food. Learn more about the functions and 3D structures of nucleic acids, proteins, and molecular machines at the RCSB Protein Database.
Okay, so now since we have a better idea about the structure of the protein, in this figure we see an example of how a protein is interacting. Protein here is interacting with the drug. Here is a protein, and these are the side chains, amino acids of the protein in blue, and this is the drug in orange, and these groups from the drug and from the amino acids of the protein are complementary to each other. They're interacting with each other. The drug should fit this binding site. It should have the proper shape, and it should also have the proper intermolecular bonds, which we will discuss soon, that represent the way these function groups are interacting with each other. Okay, so let's look at the overall process of the receptor-ligand interaction. If your receptor is a protein, it would look like that, which is a chain of amino acids. This would be the binding site, which is a hydrophobic hollow that is nearly the correct shape for the natural chemical messenger. At this surface, you will see the amino acids, which is the side chains for a protein that should be recognized by the chemical messenger. Sometimes a receptor might have another binding site that is different from the binding site for the natural chemical messenger. This binding site is not recognized by the natural chemical messenger, but it's another site that a ligand or a drug might bind to and affect the activity either positively or negatively. This binding site is referred to as allosteric binding site. The binding process starts by the binding site accepting the messenger and binding to it through the intermolecular bonds. By these intermolecular bonds, the shape of the binding site will change such that the bonds become stronger. The change in this shape in the binding site is referred to as induced fit. Notice how the bonds here got stronger and the shape of the receptor has changed from this to that here. And notice also how as a protein we have some amino acids such as serine, aspartate, and phenylalanine binding to the messenger here and making these bonds stronger. The resulting induced fit maximizes these intermolecular bonds and results into something called as the signal transduction, which is a signal that's transferred inside the cell to produce the biological effect. The signal transduction is similar to an effect called the domino effect, where you activate one protein, for example, and then this protein leads to activation of the other proteins, leading into the series of signal transductions, which leads to the biological effect. Notice that the messengers do not enter into the cell. They only bind to the surface, and then they leave the receptor unchanged and don't stay bound permanently. We should also notice that the binding sites are so specific such that any small change in the messenger can affect the induced fit, and this can affect the signal transduction and can affect therefore the biological effects. The binding interactions should be strong enough to hold the messenger into its receptor such that it produces the signal transduction and the biological effect, but it should also be weak enough to allow the messenger to depart the receptor. Therefore, there should be a fine balance between the interactions of the messenger with the receptor. It should be strong enough, but at the same time weak enough. If we design a molecule that binds to the receptor very strong but does not leave the receptor, then this molecule is gonna block the binding site, and we will have a molecule that we refer to as antagonists, which we are gonna discuss later. Now let's talk about the types of the intermolecular bonds that can occur between a ligand and its receptor. The first one is the covalent bond, which is actually not commonly used because if it binds into the receptor, it would be very hard for the ligand to leave the receptor and will produce an antagonist, as we described earlier. But it is the stronger kind of bond. The energy that it takes to break this bond ranges from 150 to 1000 kilojoule per mole, depending on the bond type. Triple bond will be stronger than double and stronger than single, and it also depends on the atoms involved in the bond. Covalent bonds are sometimes irreversible, and they are used to produce ligands that are designed to bind and not leave the receptor, such as toxins, poisons, chemotherapeutic agents like antibiotics. In this figure here, we have an example of how a covalent bond is formed. Just for example, if we have an OH group here in the binding site and we have a leaving group here in the messenger, so the binding occurs and the covalent bond here is formed. Again, this is not a commonly used bond unless you intend to have the messenger to permanently bind to the receptor. The second type of intermolecular bonds is the ionic, also known as electrostatic bond. It is the second most stronger intermolecular bond after the covalent bond we described earlier. The energy it takes to break this bond ranges from 20 to 40 kilojoule per mole, and the ionic bonds occur between chemical groups of opposite charges. For example, if you have an amino acid here with positive charge on the nitrogen, and you have another carboxylic acid here on the drug with a negative charge, then we'll know that the bond here is an ionic or electrostatic bond. The opposite can occur here. You might have the positive charge on the drug and the negative charge on the target, for example on an amino acid, and again you will have another ionic bond, another example of an ionic bond. As you can imagine, the closer the ions to each other, the stronger is the bond, and as the distance increases, the bond becomes weaker. Ionic bonds are considered the most important initial interactions as the drug enters the binding site because they have a long range of interaction. The third type of intermolecular bonds is the hydrogen bonds, which are weaker than ionic interactions, but they are stronger than van der Waals interactions, which we will see after this one. Hydrogen bonds occur between electron-rich heteroatoms such as nitrogen, oxygen, or fluorine. Nitrogen, oxygen, or fluorine, an electron-rich heteroatom, and an electron-deficient hydrogen that is attached to another heteroatom such as oxygen, nitrogen, or fluorine. The heteroatom that's attached to the hydrogen is known as the hydrogen bond donor, and the other heteroatom is known as the hydrogen bond acceptor. What's unique about this hydrogen bond is that it's directional, which means that the angle between the heteroatom, the hydrogen, and the other heteroatom should be close to 180 degrees in order for the hydrogen bond to occur. The fourth type of intermolecular bonds is the van der Waals interactions, which is the weakest among the bonds we discussed so far. It has a bond energy of 2 to 4 kilojoule per mole. Van der Waals interactions occur between hydrophobic regions of the drug and hydrophobic regions of the binding site of the receptor. The way these bonds are formed is when a transit change or when a distribution of the electron density occurs in one hydrophobic region and another disturbance and the electron density occurs in the other hydrophobic region in the binding site. These partial positive, partial negatives charges will result and attraction occurs between the hydrophobic regions. As you can imagine, these van der Waals interactions require that the hydrophobic regions be very close to each other, so they depend on the distance. If the distance is large, the van der Waals interactions will not occur. Although the van der Waals interactions are very weak, but they are very crucial to the binding process because of the cumulative effect of the overall van der Waals interactions from the drug and the binding site, from different parts of the drug and the binding site. Please differentiate between van der Waals interactions and what's called hydrophobic effect. Hydrophobic effect is what happens when nonpolar molecule aggregate in an aqueous solution in order to separate from water and minimize its exposure to the water polar molecules. Hydrophobic effect normally occurs between hydrophobic regions of the drug before van der Waals interactions take place, meaning that before the drug approaches the binding site, they have to get rid of the water molecules and they aggregate with each other. These hydrophobic regions aggregate to each other before van der Waals interactions occur. Okay, so after we have studied the most commonly used kind of interactions, there are other kinds of interactions that I'm going to list here. The first one is called ion-dipole interactions, occurs between an ion and a polar molecule. Here's the dipole. We have a partial negative, partial positive being attracted to an ion. Another example here, if you have a positive ion for an ammonium, it's attracted to this dipole, so it's called ion-dipole interactions. Now, if you have a polar molecule here as part of the receptor site, and you have another polar molecule here as the drug, then a form of bond called dipole-dipole interaction can occur, as in this example here. So the molecule will align such that the positive charge, the partial negative with the partial positive, and the partial negative with the partial positive. The third example here is for the induced dipole interactions. If you have a dipole or a polar molecule here as a drug, and you have a hydrophobic region like the benzene ring here as part of the binding site, then this dipole molecule can induce the hydrophobic region here and cause partial positive and partial negative. Similarly here, if you have an ion as part of the drug and an induced dipole and a hydrophobic group, then this ion can induce the hydrophobic group and cause what's called ion-induced dipole interactions. And it doesn't have to be the drug that's always positive and the binding site that's always have the hydrophobic region. It could be the opposite. Now let's see what impact do intermolecular bonds have on affinity and on biological activity. Well, first, affinity is how tightly the drug binds to the receptor. This means that the stronger the intermolecular bonds, the higher the affinity. And affinity is typically measured using what's called the dissociation constant, Kd, which is the concentration of the drug when 50% of the receptors are occupied. It is proportional to potency, which is the amount of drug required to produce a certain effect, and it is also proportional to efficacy, which is the ability of the drug to produce the maximum biological effect. This is just a brief overview, and you will see more details on the definition of affinity, potency, and efficacy. I just wanted to relate how the intermolecular bonds are related to the affinity. I just want to explain how these two are related. Okay, so we have seen how intermolecular bonds affect the affinity. How do they affect the biological activity? The intermolecular bonds are responsible for the induced fit that occurs to the receptor. So if we have the proper induced fit, then we will end up having the signal transduction. And the signal transduction, of course, leads to the biological effect. Now, if we need to design a drug that should produce the same biological effect, then we would design a molecule that should produce the same induced effect that the natural chemical messenger will produce, and thus leads to the signal transduction and the biological effect desired. To accomplish that, this drug or ligand should have the correct binding chemical groups, and these groups should be positioned in space correctly to interact with the complementary binding sites at the receptor, and they should have the correct shape to fit into the binding site. The ligand should bind reversibly and leave quickly. Due to all these features, it is difficult to design a drug that is very similar to the natural chemical messenger. But if we do, this drug would be very similar in structure to the natural chemical messenger, and we would call it a full agonist. In this example, in this figure, we see the same effect of the agonist compared to the effect of the natural chemical messenger we have seen in figure 5 previously. Okay, so what happens if we design a drug that produces a different induced fit? In this case, the receptor is not going to be able to produce the signal transduction and the biological effect we are looking at. And in this case, this drug will be called an antagonist. This drug would bind to the binding site and prevent the natural chemical messenger from binding to the binding site. It competes with it and will, as a result, the receptor will not be activated and will not see the full effect. Antagonists are of many kinds. Some of them bind reversibly to the binding sites, others bind irreversibly. Some of them can produce no effect at all, and others can produce partial effects. So we're gonna discuss all these kinds later on. Based on that explanation before, I'd assume you would figure out which is easier to design, an agonist or antagonist. And based on your answer, look at the drugs at your pharmacy and see what is the most common kinds of drugs. Do you see most of them as an inhibitors, blocking agents, and antagonists, or do you see most of them as agonists? Okay, so now we know the importance of these functional groups in forming the intermolecular bonds and how this affects the induced fit and leads to the signal transduction. How can we define what function groups are important? So if you have a drug like that and you have these functional groups, how do you know which function groups are important? Because once we do, the function groups that are important, we can modify them and improve the potency, the selectivity, and be able to produce safer drugs. To evaluate the importance of these functional groups, we follow a systematic fashion to determine how these function groups are important, and this way is referred to as the structure-activity relationship. We have seen this in an example in the previous session, where we have a molecule and we would change the function group, either alter it, remove it, or mask it, and then see the effect of that on the activity, and then based on the results we can decide on the important function groups. Once we know what are the important function groups, then if we identify the relative positions in space for these function groups, we define what's called a pharmacophore. So in this particular example, we modified the OH group here with this group, and the activity was reduced, which means it is important here. And another modification was to replace this function group with this, and the activity was unaffected, so we concluded that it is not important. And we repeated the same process for this group, and then we realized that these three moieties or function groups are important for activity, and if we define the relative position for them in space, we can define the pharmacophore groups for this drug. So we have seen how we defined the pharmacophoric groups in the previous example. So here this figure shows the same pharmacophoric groups we defined in the previous example. We have the oxygen, the aromatic, the benzene ring, and the nitrogen, and then if we define the relative positions of these three groups in space, we call this the 3D pharmacophore. There is another kind of a pharmacophore called the generalized bonding type pharmacophore. In this type, we define the interactions that each of this group can be responsible of. So instead of defining an oxygen here, we would say this oxygen is responsible for making a hydrogen bond acceptor with the binding site. This aromatic benzene is responsible for making van der Waals interactions. And finally, this nitrogen, the quaternary nitrogen, is responsible for making an ionic interaction. Once we define the type of interactions, again, we define the relative positions of these interactions in space. This form here is called the generalized bonding type pharmacophore. In this example here, we defined the three most common types of interactions. I mean, we used the three most common types of interactions, but it could be any other interaction that any of the other interactions that we defined earlier in the intermolecular bonds. Notice that in addition to defining the distance between these functional groups, we can also define the angle between these arrows or distances to be certain of the positions. Now, why would we bother defining the bonding type pharmacophore rather than the normal three-dimensional pharmacophore? It is because this kind, the generalized pharmacophore, is more useful in drug design and discovery. The reason is, instead of being strictly tied to the type of the atoms involved, instead of saying we should have oxygen, we should have an aromatic ring here, a benzene, and we should have nitrogen and ammonium group, I could actually say that I need a hydrogen bond acceptor here, which could be an oxygen, a nitrogen, a fluorine, and I need a function group that can make a van der Waals interaction here, which could be a benzene ring or maybe naphthalene ring. And here I need a positive ionic group. So this is more general definition that could be used to describe the pharmacophore for the active molecule. Another way of defining the pharmacophore for a number of active drugs is using the similarity principle. What does the similarity principle state is that similar biological activity, drugs that have similar biological activities and bind to the same receptor should have a common features among them. These features are defined as the pharmacophore. Let's look at this example here. We have three drugs. They have the same biological activity. They are both, all of them are painkillers, and they bind to the same receptor. This means that there must be some common features among all these three molecules, and these common features can be used to define the pharmacophore. In this case, you will see that these, the aromatic with the OH here, is occurring in all of them. The same for the nitrogen here, the quaternary or the ammonium nitrogen. You can imagine if you have more than three drugs, say you have 20 drugs, it will be difficult to find the common features, and that's when you will need the use of computer programs to perform that. Especially if you're trying to find the generalized type pharmacophore.
So now that we know what a pharmacophore is and how to define it, and we know what the pharmacophoric groups are, let's think about how can we modify these groups to optimize the features of a drug. If once we define the pharmacophoric groups for a drug, then we could actually replace some of these groups with other equivalent groups basically replace them with groups that have the same valency and have similar physicochemical properties. These groups that have the same valency and similar physicochemical properties are known as isosteres. Examples of such groups are here in this table, and these are called classical examples. And I advise you to look for non-classical examples of isosteres. Just Google it and find out what is a non-classical isosteres. These are the classical examples of isosteres. Basically, you will see these groups, for example, CH3, NH2, OH, fluorine, chlorine, and the thiol group. These could be replaced one for the other, and they have the same valency, meaning they all have they have the same number of bonds that they can be attached to an atom, or basically they can attach from one side. So they are called univalent isosteres. The same for the proline and the isopropyl group, and the iodine and the tertiary butyl group. So these actually have same valencies, and they also have similar physicochemical properties. So in this line, these could be replaceable. In this line, these two could be replaced with each other, and the same here. And there are also the bivalent isosteres. These are examples of the CH2 and HO and S. Notice that in this class, each group can be attached from both sides. I mean, it can be attached from two sides, like the CH2 here can be attached from one side and another side, and the same for the NH2, for the NH and the oxygen and the sulfur, and that's why we call them bivalent. And the same applies for this group, bivalent. They can be attached from two sides, so the valency is 2. And then the trivalent isosteres are the ones where you have 3 sides, or in this case a double bond is counted as 2, and then one here is 3, so they are trivalency. And another example of isosteres that could be replaced with one another are the ring equivalents. So for example, this benzene ring can be replaced by this pyridine ring, and the same for these groups. Notice that here you have 6-member ring that could be replaced with each other, while here you have a 5-member ring that could be replaced with each other. Anyway, so back to isosteres. Like we said, groups that are similar can be replaced for one another. Now, like I mentioned, once we define the pharmacophores, then these pharmacophoric groups we could modify one for the other in order to improve the features of the drug. Now, if we substitute one function group for the other and we retain the activity, the biological activity for that drug, then these isosteres are now called bioisosteres. So I want you to differentiate between these two terms. Basically, isosteres is a broader term, it's a more general term than bioisosteres. So bioisosteres is actually is a narrower term. Basically, if a drug is a if two groups are bioisosteres, then they are by default isosteres. But if two groups are isosteres, then they are not necessarily bioisosteres unless they retain the desired biological activity once they are substituted in a molecule. Okay. Now, why is this important? Why do we need to care about knowing the isosteres and the bioisosteres? Because if we have a drug that, for example, has a function group that is important in binding to its target receptor, but this function group is also problematic, it's responsible for some toxicity, then we could look for the isosteres and for that group and try to substitute that group with its isosteres, and then hopefully find another group that would still retain the ability to bind to the target receptor, but would not have the toxic effects. This way we would solve the problem of the toxicity and at the same time maintain the biological activity. If we succeed in finding such group, then now these two groups will be defined as bioisosteres. Okay, and this is a very important concept that is used to optimize the pharmacodynamic and pharmacokinetic properties. I hope you still remember the difference between these two properties. And it's used a lot in drug design. It is also important that the concept of bioisosteres is specific to a particular group of compounds or drugs. In other words, two function groups that are isosteres might be bioisosteres for one class of drugs, like maybe the antihypertensive drugs, okay, or the beta blockers drugs. But the same groups, isosteres, might not be bioisosteres for another class of drugs, like maybe the antihistamine. And I want you to think about it. Why is that? Why is that possible? Why would two groups that are isosteres be defined as bioisosteres for one class of drug but not the other? Okay, just something to think about. Okay, the next thing I want to talk about is the concept of prodrugs. Prodrugs are compounds that are by themselves inactive, but once they are inside the body, they are get activated most of the time by metabolism, by metabolic enzymes, such as the esterase, for example, is the most common type of activation for these prodrugs. So they are inactive outside the body, but then once they are inside the body, they are activated. Another way to explain it is if you have in vivo experiment, I hope you still remember the in vivo, which is if you have the target cells extracted and separated in a test tube, for example, then if you try to test or try to give this drug and test it on the test tubes, then it will be inactive. Now, once it's inside the body, once it's in vivo, then this drug is activated now and become active, and it's converted to the active form, and it exerts its pharmacological activity. Why do you want to have such kind of drugs? Why do you want to use prodrugs? Apparently, prodrugs are very useful actually in drug design. There are many uses for prodrugs listed here, and I picked 3 examples that I will talk about in today's lecture, but we will see all these examples, all these uses throughout the curriculum in the first, second, and third year. We'll see a lot of examples on prodrugs and their uses. Let me just give you a brief idea of the uses of prodrugs with some examples today, and then we'll remind you again each time we visit a prodrug how this is being used. So generally, the goal of having a prodrug is to solve a problem. For example, it could be a problem with the acid sensitivity, a problem with the drug not being able to cross the gut wall of the intestine, or the gut wall if taken orally, or it could be that the drug is toxic, or maybe have a bad taste or short duration of action. So we want to improve that feature. But once we improve that feature, we lose the activity of the drug. So we want to overcome this problem. So we design a prodrug that would overcome such a problem, and then at the same time, once it passes over this problem, like maybe once it passes the membrane or past the mouth in the case of the bad taste, then then you rely on the metabolism or the enzymes in the body to convert this prodrug into an active form. We'll see. We will understand better with these examples. Okay, so like I said, in the prodrugs, most of the time it relies on the use of ester groups, and therefore this ester group is hydrolyzed with esterase enzymes. But not all of them actually relies on the ester groups, rely on the esterases. Some of them could be metabolized by other enzymes, amidases, methylation, whatever. And some of them actually can rely on the activation by light. It's called, like as in the case of the photodynamic therapy, as I mentioned in the example soon. But before that, before we start with examples, let me list the uses of prodrugs. So one of the common uses is to improve the membrane permeability, which we'll see in this example here, and prolonging the activity. In the next example, we'll see that. And then another use is to mask the toxicity and side effects. So, for example, you could mask the toxicity of a drug by adding a function group, and then you would lose the activity for this drug. But then once the drug is reached to its target, then that drug would be metabolized and converted into the active form and exerts its biological activity. So this is an example we'll see throughout the curriculum, but not in today's lecture. And then you could also use prodrugs to improve the water solubility. You could increase or decrease the water solubility. Like in the example of the bad taste, some of the antibiotics like chloramphenicol has a very bad taste. So one way to overcome this problem is to design prodrug that is not water soluble. So when you take the drug, it does not dissolve, it does not reach the taste buds, and therefore you cannot taste the drug. But then once it's passed through the mouth into the stomach, the drug is converted into its original form, and now it's active, and you solve the problem of the taste, and at the same time you rely on the enzymes of your body to convert the drug back to its active form. You could also use prodrugs to improve the selectivity of the drug to its target enzyme. You could improve the chemical stability, like if the drug, for example, is metabolized by certain enzymes, then you could add a function group to protect that the drug from being metabolized by the enzyme, and then once it reaches its target, it's converted to the active form. The last example here is using a prodrug as a sleeping agent, which we'll talk about in today's lecture. So I choose 3 examples today, which is improving membrane permeability, prolonging activity, and using prodrug as sleeping agents. Okay, so let's start with the first one here. Okay, the first one here, we have a drug. It's called enalaprilat, which is a drug used for hypertension. And the problem with this drug is, if it's taken orally, the drug would be too polar. It would be ionized. It would be too polar to pass the lipophilic gut wall and be absorbed. Okay, so enalaprilat, oh, this is misspelled. Should be enalaprilat, AT, not ID. Is if the R group here is, if it's a hydrogen, then you have a carboxylic acid here, which is ionizable and highly polar, so the drug cannot pass the gut wall, it will never get absorbed. So how can we solve this problem? We substitute this R group with an ethanol. Okay, so now you'll have an ethanol group here, and it's no longer a carboxylic acid, it's an ester, which is less polar, and now the whole drug is more lipophilic. So then the drug, when taken orally, and now it's called enalapril. Okay, so when the drug is taken orally, then it has enough lipophilicity, it is more lipophilic, it could actually pass the gut wall and be absorbed into the blood. Now once it reaches the blood, then there are enzymes called esterase enzymes that will break this esterase and convert the drug back from enalapril to enalaprilat, and the drug can now exert its biological activity. So the drug is converted back to its active form. Okay, this is a common approach that's actually used a lot to improve the absorption of the drugs. Okay, for those who work in the pharmacy, you might recall seeing enalapril um, or also sold under brand name Vasotic. Um, but you probably have never heard of the name enalaprilat. For those who work in a hospital, you will hear, you can see the name enalaprilat, and it's given as an IV injection. So think about it. Why do we use enalaprilat as an IV injection, but we use enalapril for oral formulations? Okay. The second example I wanted to share with you about the uses of prodrugs is to prolong the activity of a drug. This is an example of a drug called fluphenazine, which is an antipsychotic drug used, for example, to treat schizophrenia. Okay, this drug is actually made hydrophobic by adding this fatty ester. If you look at this ester group here, so this is a carboxylic acid again, just like the one we saw in the enalaprilat. So by adding a fatty ester here, we make the drug lipid soluble. Why do we do that? We make it lipid soluble such that when we inject it into the muscle through intramuscular injection, it does not dissolve as fast. You know, the muscle is more hydrophilic. It has more hydrophilic nature. So if you give this drug, which is lipophilic, if you give it by injection into the muscle, then this drug is gonna form a depot. It's like gonna form a cluster together, and it's not gonna dissolve as fast. It will be dissolved slowly over a long period of time, and therefore this drug can be extended into the body. The activity of this drug will be extended over a longer period of time, and as soon as the drug is dissolving, as soon as it reaches the blood, it converts back by esterases enzyme. It converts back into the original form, the active form, and exerts its pharmacological effect. Okay, so this is another example of the use of a drug where we make the drug more hydrophobic, in other words, more lipophilic, and then makes it dissolve less in the muscle, and such that the drug is released slowly into the blood, and as it's released, it converts into the active form. Okay. The last example I want to share with you about the use of prodrugs is when we use them as sleeping agents. This is an example of a drug called Foscan, which is an anti-tumor medication. This drug, once it's administered to the body, it accumulates in both the normal and tumor cells. Okay. Now, if this drug is active, so the drug by itself is not active, but if it is active, then it can affect both tumor and normal cells, while we want it to affect only the tumor cell to be effective. So the drug by itself is not active. Now, once it's administered into the body, it accumulates in both cells. Now, then we make the tumor cells only exposed to the light. So we apply the light only to the tumor cells, and the light would activate the drug, and now the drug can destroy the cell. Okay, so basically this is one use of a drug that is inactive, but then it's activated by the light, and then we use that as a way to target the tumor cells selectively and avoid targeting the normal cells. Okay.